The overall goal of this procedure is to measure gene expression from reporter constructs after introducing them into Barley Aleurone cells by particle bombardment.
This method can help answer key questions in plant hormone-regulated gene expression, such as how specific signaling molecules act to regulate abscisic acid and gibberellin-induced genes. The main advantage of this technique is that it provides higher throughput than previous methods, allowing a large number of gene constructs and incubation conditions to be tested.
Demonstrating the protocol will be Grace, a student in my lab.
First, place a few embryo-less grains into the lid of a 90 mm plastic petri dish containing 20 milliliters of imbibing solution and 20 microliters of chloramphenicol. Using sterile forceps, gently remove the transparent seed coat from each grain.
Peeling the seed coats from the barley grains is a critical step. If the seed coats are not fully removed, the gold microcarriers will be prevented from entering the Aleurone cells. But if the peeling is done too roughly, the Aleurone cells can be damaged.
After peeling the seed coat from all of the grains, transfer them from the petri dish to a vermiculite plate. Then, place the peeled, embryo-less grains in an incubator at 24 degrees Celsius for 16 to 20 hours. Now, label a 1.5 milliliter tube for each treatment that will be included in the bombardment experiment. To each tube, add 2.5 micrograms of UBI luciferase internal control plasmid, 2.5 micrograms of Auguste reporter plasmid,